Welcome to the Plant Breeding and Genomics Community of Practice webinar, the development of SNP-based tetraploid maps for potato. My name is Heather Merck, and I'm the content coordinator for PBG and your host today. For those of you unfamiliar with the Plant Breeding and Genomics Community of Practice, aka PBG, I invite you to explore our training resources at www.extension.org/plant_breeding_genomics. I also invite you to subscribe to our newsletter, PBG News, to stay up to date with PBG webinars and more. During today's webinar, Dave Douches and Joe Coombs of Michigan State University will talk to us about their recent work to develop tetraploid maps for potato. Today's presentation is also available at www.extension.org/pages/63187. Dave Douches is a professor in crop and soil sciences at Michigan State University. He leads a breeding program that focuses on developing varieties for the potato industry that have improved processing quality as well as disease and insect resistance. Joe Coombs has been working with the potato breeding program since 1996 as an assistant breeder focused on the program's breeding and statistical analysis. I'll now turn things over to you, Dave and Joe.
Thank you, Heather and John, for hosting the PBG webinar today, and we're happy to be here. And this is a special occasion because this is the first webinar being done from our new Molecular Plant Sciences edition at Michigan State University.
And hello, I'm Joe Coombs, and we're going to go ahead and get right into talking about the development of SNP-based tetraploid maps for potato.
Thanks, Joe. So what we want to do today is talk about some of the work that we've done to use the SOCAP-based SNPs and how we're using it to develop a tetraploid map in potato. And this is really a team project of, that involves a large number of people, including myself and Joe, with people like Kim Felcher, Dan Zarka, Alan Van Dynes, John Hamilton, Candy Hansey, and others, and Robin Buell. Sorry about that, Robin. And so the objective of today's webinar, hopefully by the time we get to the end of it today, is that we want you to be exposed to visualizing SNPs in the Genome Studio software developed by Illumina, including the 5-cluster calling that was done here in the SOCAP project. Secondly, to how we're filtering the SNPs to do the tetraploid mapping work. Thirdly, and how we're generating the map using Tetraploid Map software that was developed in Scotland, and then how we're initiating the QTL analysis using both single marker ANOVAs and also Tetraploid Map. As many of you know, if you come from a potato background, there have been a number of linkage maps developed in potato going back to 1988, and some of these or most of these maps are done at the diploid level, and I would say that's because of the simplicity of making maps at that level than the tetraploid level. And so you can find a number of them in the literature, and we have one that was just recently accepted that's a SNP-based one by Felcher et al. Oops, my slide jumped ahead there. So with SOCAP, one of our goals is to develop a genome-wide set of SNP markers, and under that work, there's a number of activities that we're doing on the potato side of the project. And one is we wanted to really assess the concordance between the map location of the SNPs and the potato genome sequence location based upon the pseudomolecules that's part of the potato web browser. And so that's the Felcher et al. 2012 article that was just accepted in +1. What we also want to do and what we're focusing on today is using this Infinium 8303 potato array to genotype tetraploid populations and to generate a SNP-based genetic map. And we're also going to touch upon our initial efforts for QTL analysis in the tetraploid populations using the tetraploid map. And we're also in the project, but not going to talk about today, is the association analysis of the potato diversity panel, and that's being headed up by Candy Hansey in Robin Buell's program. The diploid map that was generated was really based upon two maps. One we refer to as DRH, which was a cross between DM, the doubled monoploid, and RH, a Dutch diploid clone that was also used in the genome sequencing. D84 is a cross between DM and 84SG22, a diploid from our breeding program that seemed to have a high level of SNP polymorphism. Just jumping to the results of this is that we were able to, with this SNP mapping at the diploid level, to show that the SNPs were distributed well throughout the chromosomes and across the genome, as you can see here in chromosomes 1 to 6 and 7 to 12. If you look at it from a table perspective, what we have here is, you can see, I'll use my cursor here, that in the column under DRH, you can see the number of SNPs that we were able to map as 1,960, and in the D84 we were able to map 2,454. With 787 common, we were able to map 3,627 different SNPs back to the, and compare that to the genome browser position. If we jump over to the right side of the table here, you can see the estimated map size for the two different populations. And it's interesting that we have about a 30% reduction in the population size in D84 from DRH. However, when we look at it from a pseudomolecule perspective, that the coverage across all the pseudomolecule, the 12 pseudomolecules, is that we have 634 and 642 megabases covered with our SNPs, and we know that there's about 650 megabases in the 12 pseudomolecules.